this. So thank you for joining us in this last talk in a series. So today the speaker is Dr. Hiba Sheheitli, who is a computational neuroscientist at the Institut de Neurosciences des Systèmes, ex-Marseille University. She holds a PhD in theoretical and applied mechanics from Cornell University with emphasis in applied mathematics. Before joining INS, she was an assistant professor in mechanical engineering at LAU, where her research addressed novel nonlinear interactions in 3D rigid body dynamics. Her current research focuses on dynamical systems characterization and modeling of whole brain nonlinear dynamics in the context of brain disorders. And she's an active member of the EU Human Brain Project and eBrain research infrastructure. She will be talking today about the virtual brain. Thank you for being with us. Hiba, the floor is yours.
Yes, hello, Fatima. Thank you for the kind interaction and for the invitation. Do you see my screen?
Not yet.
Okay. And now in presenter mode, or do you see only the first slide?
We see the first slide, yes.
Okay, so you see the first slide full screen?
Yes.
Not the next slide?
No.
Okay, good. Because I have presenter mode and then. Okay, good afternoon, everyone. Let's start. So really today what the title is the virtual brain from dynamical systems to epilepsy surgery. And it's on data-driven personalized brain models for translational medicine. I'm still struggling with this camera. I'm not sure where to put it. Okay, this should be okay. So what I really want to do today is introduce the virtual brain, what it is, what is it, and the building blocks, the concepts behind it, and then show how it's the first success story per se, how it's been used in a clinical trial for epilepsy surgery. Okay, so this is work that's been done in Victor Girsa's group, which I'm a member of. It's really more than a decade's work, and it's not the most recent per se, and most of it is done by the team, not by me personally, even though I'm part of it, of the let's say the main team behind the virtual brain. The more current work now is on more new other diseases such as Alzheimer's, Parkinson's, and so on. But I thought as a first contact with the CAMS community, I will start by really giving the concepts and the most, let's say, iconic story for virtual brain. And I'm going to keep it a bit high level. So I'm going to give the context, the framework. I'm going to, at some point, maybe I'll throw a lot of math at you, but it's really just to give the flavor of the math involved. I would not really go into a lot of mathematical derivations and so on. And yeah, I hope it will open up a discussion. So epilepsy, when I say epilepsy here, we're mainly, there's a dozen types of epilepsies. We're talking about drug-resistant focal epilepsies, okay? So 1% of the world population actually suffers from epilepsy, and almost 60% will do okay with medication. These seizures will be controlled, but the rest will have to undergo surgery as a last resort. And this kind of surgery has been done since decades now, and even though the technology and the science has advanced a lot, the success rate has not caught up, let's say, to the advancement. we still have not it's a significant amount of patients that go through surgery and then seizures would occur again. Now, the main idea of the surgery is that so a seizure is an overactivity of brain of neuronal discharges in the brain. Okay, focal epilepsy will start somewhere and then spread to other areas. And when the medication doesn't work, the surgeons, the neurologists have to identify where is this over heightened activity happening. And then they have to cut and go in there and remove that, let's say, bad piece that's initiating the seizure. Okay. And it's a challenge to actually identify this region because the brain is complex and it's not always that localized. But first of all, let me show you this is a patient here in Marseille, and there you can see that this is what a temporal lobe seizure looks like. So these are all involuntary vocalizations and movements. And this patient actually has electrodes implanted in her head. And now the seizure has ended. Okay, so when a patient is candidate for surgery, the clinical procedure is that they get SEEG electrodes implanted in the brain, and then they are monitored, and you could see the discharges occurring successively in the different regions, and that's what the doctor uses to actually identify the seizure. So if you look closely at the actual neuronal measurements that are being recorded by these invasive electrodes, you can see that you will get a sudden spontaneous increase in discharges from a baseline, and that spreads across the different measuring sites per se. So what we're seeing here is really the SEEG invasive measurements. And in that small schematic down here, you could see the different electrodes are corresponding to different areas where the activity is spreading in the brain. Okay, and the heightened activity in different areas correspond to these involuntary actions or voices that are being generated. Okay, so this is sort of to give you an overview for really the medical challenge, the medical context. So the role that the modeling and the mathematicians have been playing, that we are trying to do, is that the surgeons so far, the way to identify, and the challenge is we want to identify, we want to know really which region is the epileptogenic zone, which is the candidate to be resected in surgery. And there's a lot of statistical measures that the doctors use. Some of it is quantitative, others is really by looking. Of course, there's the symptoms, there's a clinical exam. So medical doctors have whole systematic ways to actually find it, but it's really more phenomenological and empirical. So what we want to do is we want to provide a mechanistic base modeling approach to actually identify this. And the strategy is as follows. What we need, the ingredients that we need, is first of all that we will use non-invasive imaging that's coming from MRI to actually capture the geometry of the brain. We're also going to look at the connections in the brain from diffusion tensor imaging. We're going to use that to build the skeleton for what we will call the virtual brain, the avatar. We're going to fit it, personalize it to the specific patient, and that through simulations and inference, we're going to provide the doctors with informative metrics on what our guess is for where in the brain the seizure is starting. And so this is basically the workflow. Okay. We take the imaging, we have to match it with mathematics, we have to fit it, and then we have to go back to the surgery and see if the outcome is correct. Okay. So this is all, people maybe are not familiar in the field, these are all big words and it's still ambiguous. So I will try to sort of break it down and see what all these ingredients are. And what do I mean really when I say a virtual brain avatar? Okay. So the models that we are using here, I mean, there's a lot of models, there's statistical models, machine learning models, different kinds, but here we're using a specific approach where we are going to look at the brain as a network of nodes that are representing the dynamics of the different areas. And that's the word connectome-based whole brain network models. By connectome, meaning connectome is the term to refer to the actual physical connectivities, the tracks in the white matter tracks in the brain. Okay. And the first assumption of connectome brain modeling is that even though the brain is a continuum and you have billions of neurons there, we're going to think of it as being parcellated into a finite number of regions. And of course, this parcellation are physiological, they come from histology, from anatomy. So we're not the first ones to do this. So we take the well-known atlases of the brain, the parcellations, we are going to say, okay, now the brain is going to be thought of as, let's say, 80 pieces. Each of these pieces is actually containing millions and millions of neurons. But we are not going to try to model the activity of every neuron. We're going to use mean field models. We're going to use tools from physics and mathematics to come up with low-dimensional mathematical equations that are going to capture the sort of average or mean field activity of these millions of neurons in one region. And this is a whole field in itself, mean field modeling of neuronal populations. So here, I'm just going to put one slide to give you what's a flavor of what's usually done in the field. You would, so you could, this is here just an example. Add X is one type of equations to represent the activity of a single neuron, sort of Hodgkin-Huxley type, where people are familiar with it. And then if you actually simulate the activity of, let's say, thousands of neurons by representing the activity of each of them, you could sort of plot the results as this raster plot here. Here, blue corresponds to excitatory population. Red is an inhibitory population. So some neurons, when they talk to others, they excite them, and others they inhibit them. And now if I were to say, okay, what is the average sort of activity here, and I plot it as a time series, I could see something like that. And then the idea is that we want to use mean field methods to actually, instead of keeping track of the single spikes, we just want to find an approximation for the mean firing rate. How often is this population firing per second on average? And what we would hope to capture is that we would capture qualitative changes in the state of the network instead of trying to track each of these neurons. So, for example, in this over here, that would be an awake-like state. The neurons are excited. If you go to a sleep-like state, the population will actually go into a slow oscillation of on, off, and so on. And here I'm showing just one known example from the literature. You can see that the mean field can actually capture this slow dynamics of the population while leaving out the faster fluctuations per se. So this is the first step in building the virtual brain. We have to think of the brain as parcellated into finite populations, and we need to find a simple enough reduced order mathematical model to represent the mean dynamics of the population. Okay? And once we have that, we build the network. Okay, so here, this is if you want, the most general way to represent psi. Here is the sort of the field, the neuronal activity. It's going to have, and here it's intrinsic dynamics of each of these populations. And then there's going to be local dynamics, maybe some kind of diffusion-like or synaptic interactions between neighbors, but also there's going to be global dynamics, which is going to come from the connectivity, the white matter connectivity across areas. And of course, in that case, you put in some delay because the signals need a finite time to arrive from one place to another. So before going into more mathematics, I'm going to actually give you an overview of how what the virtual brain platform is. So the virtual brain platform is an open source neuroinformatic platforms that allows you to actually do all that without really having to go through the details of the math or the programming for the integration and so on. Okay, it's open source. You can download it. There's a GUI that you could use. You choose. There are typical, let's say, or sample parcellations. You choose sample tractography for the connectivity between these regions. You choose the actual dynamical model that you want to use to represent each region. There are several ones that are known in the literature that are already implemented there. And then you can actually simulate. And you could say, you could do a sort of an in silico experimentation platform. You can say, what will happen if these connections were damaged? Or what will happen if I actually do surgery and I resect this region and so on? And what's convenient is that you could actually simulate the activity of, is the video playing? Yes. You could simulate the activity of the brain regions, but you could also generate synthetic sensor data. So you can say, if I put in this connectivity in this model, and I assume I'm virtually measuring EEG or MEG or bold fMRI, what will the signal be? So there's a forward model to actually map the source neuronal activity onto the typical neuronal sensors that are used in the medical field. So, okay, so this is TVB in general, and it's not specific to epilepsy. You could use it to model any specific context that you want to study. But now we want to use it to actually specialize it, make it most efficient for the epilepsy case. Okay, so the question is, which dynamical model do we use for the nodes? Should we go to a generic mean field model? Should we assume, take the generic neuronal population and then see how you could generate the seizures from that? That is one way to do it, but instead, so let me go through this here. Instead, for the specific case of epilepsy, because epilepsy has this very striking dynamical feature, which is the seizure, what Victor Gisa did 10 years ago is he actually proposed to use dynamical systems theory to build a phenomenological model to capture the dynamics that is not directly derived from the biophysics of Hodgkin-Huxley type equations, but will actually exhibit the canonical dynamical behavior of what seizure dynamics is. And so here I'm showing you local field potential measurements from the hippocampus of a rat. Okay, that was actually induced in the lab to be exhibiting seizures continuously. Okay, so you could see that really a seizure is kind of a instability from rest into fast discharges. Okay, and then there's an evolution of these fast discharges. And when a mathematician, dynamical system person looks at that, you can say, okay, we know how to model this kind of dynamical behavior, because this is generic of what's known as slow fast systems. Okay. And so here, if you want to extract the canonical form, you could actually model this kind of dynamics using a two-dimensional or two subsystem dynamical unit, one for fast spiking and the other for slow modulation that takes you in and out of the fast spiking. Okay, so the clean image in the upper right corner would correspond to such a canonical mathematical model. Okay. While over here, this would be the actual experimental signal. When you plot that in state space, you can see that the actual qualitative state space dynamics matches that experimentally, even though it's much cleaner because we've sort of abstracted the core skeleton of the dynamics. And if you zoom in closer, you will see that really the actual spikes, the changes in the amplitude and the interspike interval could be different when you're entering the fast discharge or when you're exiting. And these kind of qualitative features are sort of consistent even across species. So when you look at a seizure event and you're trying to see the geometry of what's happening in state space, you see something that's persistent across species and individuals. And that was the inspiration for the work that devised what would be known as the Epilepsy model that was put forth in these publications a while ago. And now I'm going to do a small detour into the math that's involved in actually building this Epilepsy model. Okay. So as we said, there's a qualitative feature on how the amplitude and the frequency of the discharges evolve as you enter and exit the seizure event. And for people who are familiar with the dynamical systems, that corresponds to actually a specific type of bifurcation. Okay, so the idea is that we can build a taxonomy of seizures. Just like if you have the periodic table and you want to say, okay, this molecule is made up of this much carbon and this much hydrogen and this much oxygen. Over here, we're going to say you have this table of we know from the mathematics that you can only go from let's say a quiet state to an oscillatory state through a finite number of mathematical routes per se. Okay, and these canonical types of entry and exit into oscillation, okay, if we use them as sort of the elements, then every pair of entry exit is going to be referred to as a type of seizure. Okay. So here, for people not familiar with bifurcation theory, I'm just putting it here to give a flavor, but it's we can discuss it later, but don't worry about the actual specificities of these. I'll try to make it more intuitive in a bit. As an example, this is a typical seizure recording, and Okay, but how are they different, these typical canonical ways of entering and exiting oscillation? Why are they relevant? It's not just name calling, okay? We can identify them because from the mathematics, we know that if this bifurcation occurs, that's what it means is that it's there's a certain relationship between or feature characteristic of the amplitudes and evolution of amplitude and frequency. So, for example, a saddle node bifurcation would correspond to the oscillations appearing suddenly with a fixed amplitude and a fixed frequency. On the other hand, you could, if you have a supercritical Hopf bifurcation, that would be you have a gradual increase in the amplitude of the oscillation, but the frequency is remaining constant as you're entering the seizure. Okay, and so on. So it's really about the dynamical signature of how the instability is happening and ending. And when this was hypothesized, that really seizures can be classified according to a dynamical bifurcation taxonomy, it had to be put through the test. And in this paper by Saggio et al., what they did is actually they looked at over 100 seizure events from recordings of more than 130 epilepsy patients, and they tried to actually classify these entry-exit events. And this is here a typical real recording of seizures. And it was found that there's really, it's not originally, there is a dominant type that almost, you can see here, I'm not sure if this is small, but for example, 41% of onset bifurcations can actually be classified as a saddle node bifurcation, that the relationship between amplitude and frequency as the seizure is starting is following this mathematical feature that we know from abstract mathematics. Okay, at the same time, the offset bifurcation seemed that there's 41%, that's a supercritical or a subcritical here, SNCC bifurcation. Okay, so you hypothesize from mathematics by looking at the canonical form, you look at the actual recordings from seizures, and you see that there is a dominant type of how seizures are happening. And so what the next step is that, okay, so we're going to make a bold move, and we're going to we're going to construct a model that can actually, when driven in a specific way, can actually undergo this kind of dynamical signature of how it goes into and out of oscillations. And so taking that predominant class of seizure-like events that was found in empirical data, happened to be the fold homoclinic class. Basically, you can see that the building blocks for seizure-like events is really fast discharges and a spike and wave event. So we put that in mathematics. We know how to use very simple mathematical equations to actually create this behavior, and we end up with 5D equations. So I'm not going to go into the details of the math now. This is just to give an idea of what it looks like. But the thing is that there's a slow permittivity variable that will, when varied slowly, will actually allow the system, these 5 equations, to go in and out of a seizure. Okay. And that can be seen in a bifurcation diagram here, where you have, as you vary Z, okay, you go from an interval of here being stable to oscillations and back. Okay. But is this realistic is there anything like that in the actual physiology? Is there a slow permittivity variable per se? And there's actually some candidates for such variables that could be at play on the cellular level. Okay, so over here, this is a recording of a seizure-like event. And if you look at the some ionic currents here that are changing slowly, you will see that they're actually playing the role of such a slow permittivity variable. So this is not the one-to-one mapping, but to say that these slow biophysical ionic mechanisms do exist and are known to be affected in epilepsy disorders. So now we found a candidate. We created a phenomenological, let's say, a minimal model to use as the dynamics for this individual nodes for the parcellation of the brain. We can go ahead and actually build the virtual epileptic brain. Okay? And the process is as such: you parcellate, you put the nodes, you put the connectivity, and then you can actually simulate what should I see on the SEEG recordings by using the forward model also. So this is what the simulation of the virtual epileptic patient would look like. So what we did here is we used this minimal epileptor model. We used the typical connectivity from the diffusion MRI of a patient. And then we, by hand, we pick some regions to be more prone to having spontaneous discharges than others. Okay, so one thing I should mention is that the epileptor model has one parameter. And when that value of that parameter is below a certain threshold, the equations will not tend to go in and out of the bifurcation. They will have, you will be in the region where you have a stable fixed point. But when you're above that, when that parameter is above that threshold, that means that this region is going to spontaneously go through these seizure-like events. Okay? If you're close to that threshold, if that region is close to that threshold, that means that it's easy when it receives input from other regions to actually go into that seizure. So it can propagate it. Okay? So in order to do the simulations, we have to make a hypothesis about this, the parameters for each of the regions in the brain. Okay? A healthy brain would have all of the regions actually with that parameter below the seizure threshold. Okay? And an epileptic brain will have a few regions with that parameter very close to that threshold, so that any noise or coupling from other regions could actually kick the node into a seizure, and then that would propagate. And this is what happened here. Now, we want to actually use it to infer for a given patient which brain region is actually that weak link, which has a parameter that's making it the first to actually go into a seizure. And to do that, we use a Bayesian inference framework. So we use the connectivity of a specific patient, and then we use the actual recordings of the SEEG recordings for that patient to do Bayesian inference of saying, okay, I assume that each brain region can be described by the epileptor equations. What value of parameter does the model have to use at each of these nodes in order to produce the SEEG signals that I am actually seeing? This is what Bayesian inference is doing. And why are we using Bayesian inference and not some other statistical model inversion techniques? It's because of degeneracy. Because the brain is complex and nonlinear, it can actually exhibit the same behavior using a multiplicity of different parameter configurations. So we do not want to start by assuming a, let's say, Gaussian distribution on all of the variables before we try to infer them. And so using a Monte Carlo, Hamiltonian Monte Carlo sampling, we can actually avoid that in the Bayesian inference. We can avoid having, for example, over here, if the actual distribution is the blue one, if I assume a Gaussian, it's going to give me some value. If I force a Gaussian distribution, it will give me one value that lies between, let's say, these two possible values. Again, here this is there's a lot to be said here. I'm just giving a high-level description, and then if anyone wants to hear more, I'll be happy to elaborate at the end. So this is what we do. Assuming this form of the equations, we fix all parameters, and then this parameter x0 here is what we want to infer for each of the brain regions. Okay, so some of them will be stable, and others will have the value that actually corresponds to oscillations. When we fit the parameters actually to, so this is a sample personalized virtual epileptic patient model, and now we can simulate again and see. So this is basically the virtual avatar of a specific patient after we have actually fit the parameters. And over here, you can see that TVB will give you the source signal, but this is a phenomenological model. So these X, Y, Z, they don't correspond to anything. There's no one-to-one mapping to, let's say, membrane voltages or firing rates. So it's only useful when we actually map it to a forward model and we say, this is the resulting SEEG. This is, and you can see here, this is when the seizure started, and then it propagated to other regions in the brain. And the goal is so the doctors already have a way to figure out which is the candidate epileptogenic region. And we want to see if, how does that compare to the predictions of the virtual brain? And this is already happening in a large clinical trial. So I'll come back to this picture later. This is already happening in a large clinical trial called EPINOV. It's a multiple center trial across France. There's 13 clinical centers involved. And the process is there's data being acquired, the preclinical process for the patients that are candidates for surgery. And you can only do this with patients candidates for surgery because SEEG recordings are invasive, and you only have that if the patient needs to go through surgery. This data is being centralized and sent to Marseille here, where we do the personalization. Then we create clinical reports to give to the medical team, and that's it. We don't do more than that. And then there are the controls, are the patients where the medical team does not receive the clinical report from the virtual brain, and the actual patients are, the subjects are the ones where the medical team has in their hands also the report from the virtual brain. And there's some, the clinical trial is still ongoing. There's some preliminary results. And since this is sort of, you only do the surgery once, so you can't really try and see if it's, your prediction, if virtual brain is doing better than the empirical approach or not. But of course, there's a lot of complexity. There's, you have some physiological rhythms that could have these fast oscillations that are not seizures. You have variability in the seizure classes, as we said, and so it's not a deterministic process, but the preliminary results are showing that for the patients where the surgery actually was successful, these patients became seizure-free after the surgery. The VEP, the virtual brain, is matching what the doctors recommended. Okay, but for the patients where the surgery was not successful, so the doctor made a judgment that this region is the epileptogenic zone. They resected this region, but the seizures remained. In these cases, it seems that the virtual brain is actually not matching what the doctor said. So in a sense, it's saying that the virtual brain is saying something different, okay? Which is a good thing in a way. We don't know if it's different and correct, but at least it's saying something different. So, so this is just some numbers on what I said. Of course, there's a battery of metrics to assess the goodness of the recommendations. But here, so we have 12 seizure-free patients where you're actually comparing against the post-surgical MRI, and you see very good match between the virtual brain and the actual result. For the non-seizure-free patients, it's not matching. So that's good. The clinical trial is still ongoing, and there's almost like 260 patients included, and we will have to wait a couple of, I think, end of this year to have the full report. And so basically I will stop here, and this is the names of all the working team that, let's say, been involved in all of this. And thank you. We take questions.
Thank you very much, Hiba, for this amazing illustration of all the work that has been done so far. So it's time for questions. Please, if you have questions, either raise your hand and unmute yourself, or you can also type in the chat.
Should I
any questions?
Stop screen sharing?
Yes. No, it's fine. You can keep screen sharing. So we have a question from Joseph Bakarji. If I'm not mistaken. Hi.
Thank you, Hiba, for this great talk. It was really interesting. I just have a couple of questions. My background is not in neuroscience, but I'm very interested in the subject. So first, when you come up with a mean field model based on these different brain regions, you're sort of doing some kind of coarse graining, right, to get like, some large reduced order network where you can do dynamics. I was wondering if is it known that there is like a separation of scales that allows you to do that? Are there like lower scales are between the neural level and the sort of region level that might be also important in the dynamics of the seizure?
Yes, timescale separation, that's a core hypothesis, the assumption of the epileptor model. But are you referring to the more general biophysical mean field models, or to the actual phenomenological epileptor that we cooked specifically for the epilepsy?
I'm referring to brain modeling in general, I guess, not necessarily for epilepsy, but I'm wondering if it's known for epilepsy that lower scales don't really make a difference, like, because you came up with a model with that assumption. So is that going to be so I guess you take into account faster scales, right? So you have slow dynamics and fast dynamics, but your model is only operating at one scale. It's not like the network is not explicitly taking care of smaller scale dynamics, right?
So first of all, let me talk about the mean field models in general. So when it's neuronal mass models, or also known as mean field models, over there, there's not an assumption of separation of timescales per se, but there's an assumption that, as you said, coarse graining. So I'm going from a system of a very large number of fast spiking neurons. And I'm going to say that when I go and when I want to go, so there's the micro scale, the mesoscale, and the macro scale. That's how we talk about it.
Yeah.
You go from, if you're interested in whole brain modeling, the way we do it, we want to, really the interesting feature we're trying to understand is on a macro scale. And we want to describe mathematically mesoscopic effects. We don't want to keep spiking neurons when we go to model the whole brain. So we try to coarse grain from the micro to the meso. And over there, we say we're gonna give up information about what each neuron is doing on the single spike time scale. And instead, we're going to say that we will be good on the macro scale if we only keep the slow in a sense of when compared to micro, but it's still fast when compared to the macro.
Mm-hmm.
So we're saying that I have millions of neurons in the, let's say, the I don't know, the temporal lobe or the motor cortex or whatever, but I'm going to say that when studying what's happening in the whole brain EEG or fMRI, I only care about how many spikes per second as an average are these millions firing, and is that constant or not, that firing rate? So I don't care who is firing or what. So if you see in the mean field models, you keep the envelope of the mean firing rate, but if you compute the mean firing rate of the microscopic system, it's not a smooth envelope. It's actually, you have these faster fluctuation in the mean firing rate, and this is what we're losing when we go to the mesoscopic description. Now, in the phenomenological modeling, we don't even care what the individual microscopic elements are. We say, I have this time series, and I have this geometry in state space and the evolution. And I know from mathematics that if I wanted 3 or 4 or 5D equations, 5 equations, to actually be able to exhibit these dynamical features, okay, I need to assume a timescale separation within the subunit. And that's why we had a fast subsystem, which is able to give me the fast discharges, and the slow subsystem, which is able to actually go in and out. And this is on a mesoscopic lens, okay? So I'm assuming that the mesoscopic dynamics actually have 2 timescales.
Okay. Yeah, makes sense.
Yeah.
All right, thanks. Yeah, I have another question, but maybe I'll let someone else ask first.
Sure.
So please go ahead. I don't see any raised so far.
Okay, so just concerning the model, when you're doing parameter estimation using a Bayesian framework, I didn't get exactly which parameters you're trying to estimate. Can we get to that slide?
Yes, sure. Let me show. Oh, I have to go to screen sharing again. Sorry, I'm a theoretician and not very. Here it is. Ah, we're still screen sharing, right?
Yeah, we're still screen sharing.
Okay. Did we lose the screen sharing?
No, it's fine.
Okay.
I see it at my end.
Okay, so let me know if you. Okay. You see the equations now?
Yeah.
Okay, cool. So this x0 here in the small permittivity variable.
Probably
okay.
if you make it full screen at your end,
Okay.
reflector mode or something like that.
So x0 is your only parameter?
I don't know what you're seeing now. You're still at the slide?
No, we're not beginning.
You're navigating. Yes, sorry. Bear with me. I'll get there. Okay. I think closer. Yes, here. X0 over here in the z dot equation.
Yeah. Okay.
But it's x0 for each region.
Right.
So if
but that's it. That's the only parameter.
Yes, the rest is actually tuned and very smartly devised, but not really magic. It's sort of, you just take the canonical forms of these bifurcations, and then there was some tweaking to actually get the permittivity variable to take you in and out. And then you allow, then you can see that when you have that epileptor, it's the x0 that takes you from a regime of where you have stable fixed point, or you are continuously seizing.
Okay. Yeah. So I was wondering, is it possible that you can get a different model, because that's not a unique model, right? You can have a different model that exhibits the same sort of bifurcations. And like maybe assuming, because it's a polynomial model, and so I've worked on sort of discovering polynomial differential equations from data, and I wonder if that would work in your case, where you have data for your, I guess, your EEG data, and from that maybe you can infer the dynamics. But in that case, you would have many more parameters, and I don't know if it would be necessary or not. Like maybe you can get a sparser model, or have you thought about, maybe improving that model, or you think that's just good enough? You don't need
No, definitely. There's a lot of you could also fit a much simpler, let's say what's called like there's a lot of, like, you could assume you could choose whatever form to start from, right? The question is, how much can you actually simplify the model inversion problem? So how much can you put in the model already? What do you know? That's the thing that, for example, we're developing the Parkinsonian brain right now, right? In Parkinson's, we don't have that striking dynamical feature, which is the seizure.
Yeah.
Where we can actually take that and already constrain our model. So we start with a generic mean field model population, just assuming that populations are spiking neurons, and we use a reduced mean field model there. And then we will have to pick, you could leave one of the parameters, let's say a variable, and then you will have to fit, and you could do Bayesian inference or other kind of techniques, right? But in the epileptor, we're lucky in a sense, because it was found that the neuronal signals actually exhibit the signatures of canonical bifurcations. And so we already have that. I don't know if you're familiar, there's a book by Ezekevich called Dynamical Systems in Neuroscience, where he showed actually that, so the epileptor here is not wasn't cooked from scratch. It was taken from canonical models for spiking neurons already. So there's a lot of work that shows you that, because spiking neurons also on a single neuron level actually show those dynamical features of canonical bifurcations, and different types of cells can be modeled using different canonical bifurcation equations. So the hippocampal cells, you would use this canonical form, the thalamic cell, the pyramidal cell, you would this one. So this had already been done in the field. It was showed that you could take, minimal canonical forms and capture qualitative features of single neurons. And so what was done here, it was said, oh, look, in a seizure, you have a population of neurons that's exhibiting this kind of dynamics that's usually exhibited by, let's say, simple neuron. However, it's a bit more complex because it's spontaneous and you go in and out of seizure, and there's variability in the seizure types. So this required an extra, let's say, design step to actually say, how can I mix and match the actual canonical bifurcation types and then leave just something to fit? And this X0 is convenient for that because it actually can be interpreted as the epileptogenicity of a region. Because if it's low, then it's not epileptogenic. If it's high, then it's spontaneous. And if you're close, then you're sort of in a candidate to be in the propagation zone, because if kicked, you could go. So it's not unique. It's not, and it's polynomial, as you said. You could maybe simplify it, make it nicer, cleaner if you, let's say, start a priori, assume polynomial fit, but it works as such, let's say.
Yeah, nice. Yeah, makes sense. That's really interesting also that I guess in the case of epilepsy, regions behave like individual neurons. Is that the case because in epilepsy you have some kind of synchronization? And
yes. It's not that they behave like single neurons, but they exhibit these transition into oscillations, let's say, right? You have these, the idea is that when you look at EEG or SEEG, a seizure is really a fast oscillation. So in a sense, you have an increase in amplitude of something oscillating, and it's sort of like the birth of a limit cycle, right? And usually when you have spontaneous neurons, that's what happens. But a seizure is actually a pathological increase in synchronization, exactly that. So that's why the signal adds up and you actually see it on EEG. Otherwise, the EEG is supposed to be very low amplitude. It's supposed to see these big things, and it's when you get this pathological synchrony that the signal amplifies. Yes, it's exactly that.
I see. Yeah, nice, makes sense. Thanks.
Let me ask one question, Hiba. When you said at the point where you introduced that slow permittivity variable, so it was a variable introduced by the model and then suggested by the model, and then you're trying to understand what is the physiological meaning of that. So this is something that the model is suggesting, and then you're trying to, not the opposite. It's not that the model is trying to replicate the existence of some parameter through that Z or some value.
Yes, the model is trying to give you the possibility to go from a quiet state to fast discharges and then back out, and on the entry and the exit to have a different frequency amplitude characteristic. This is what the model is trying to do. It's trying to give you that the minimum ingredients you need to go to exhibit that. And it appears that you could do that if you assume a slow fast system with a parameter that can vary slowly. But now, so when you studied cooked up the epileptor, Z was assumed to be an actual parameter that you could vary by hand. And now we want to allow that to be an internal variable that actually can be driven by the internal dynamics of the region, but also by its coupling to the other regions on the network. If you see, it's simply a linear, Z dot is linear in the slow variable, in the X1. So it's really not adding, let's say, extra nonlinearity or something. It's just giving you this evolution. And then we introduce a parameter in there, because if x0 is below a certain number or above a certain number, no matter what x1 does, you will never go into a seizure, and vice versa. So, but then it's because we know that there is multiple time scales. When you actually go, if you want to model detailed biophysical neuronal single neuron models, you do have these time scales. We know that there are ionic currents that are operating on multiple time scales. There are synaptic dynamics that are operating on, so you can go 1 millisecond, 10 milliseconds to 100 milliseconds. So we have this order of magnitude difference in the physiological processes. So it was easy to find candidate, let's say, processes once the math suggested that you need such a, let's say, player to actually get this variation. That's it.
Thank you for explaining that. Well, any other questions from the audience?
Yeah, I have a small question.
Please go ahead.
Concerning your system in X1, Y1, Z, X2, Y2, which is a system of ODE, I'm interested in the mathematical and computational aspects. So this is a highly nonlinear system. So how do you solve it? What are kind of software? How do you solve that system? Can you say something about it?
Yes, of course. We need it to be highly nonlinear, and we don't. Of course, we don't solve it analytically. But what we do is,
I mean, I'm numeric. Yeah. I mean, simulation.
Yes, this is already. This is already implemented in the virtual brain, which is a Python-based platform.
Okay.
So everything is built in Python. It's now it's open source. So if you wanted to use this, you don't even need to code. You can go and just download the virtual brain, and it will be running Python-based numerical integration, basically. You could also, and what we do is, in order to really understand what it can do for the one node, so one epileptor, there's already full bifurcation analysis. So without solving for individual solutions, we know, given parameter values, the full range of qualitative types of solutions that we can get. And so we can tune our models accordingly. And then when we want to actually couple it in a large brain network, so we have, let's say, 80 or 100 or 400 of these, so 400 times 5 equations coupled on the network, we don't do full bifurcation analysis. We actually fit to individual patients. So we fix parameters, and then we numerically solve it numerically to actually find the time series solutions. And that's also done through Python-based numerical simulation.
Yeah. But mathematically speaking, you have, let's say, if you take the fourth equation here, you have a differential and integral equation, because the G satisfies also it satisfies an integral equation. So anything done about the mathematics for that system? This seems to be interesting from a mathematical angle. Are there references on that, or?
Yes, so the epileptor is already been published and studied and analyzed. But do you have any specific, something specific in mind that you are thinking? Like,
no, I am curious about the mathematics, and it's interesting, just. As a model, I mean, I think it has a lot of originality to be a highly coupled system with integral differential system. So So I mean,
Yes, you're fine.
it seems that what you're saying is that this is something well known.
No, this is what's well known is the epileptor is not well known. Epileptor is known by people who are interested in doing, let's say, reduced whole brain epilepsy models. They go to the epileptor. What's well known is the mathematics that inspired the epileptor. So what's well known is that, for example, you could use a saddle node bifurcation canonical type of equation to, let's say, so this thing here, the x dot equal x minus x cubed, the quadratic with a slow variable, for example, is known as the Ezekiel-Fitch model, and it's known to be the minimal 2D equation to give you many types of spiking neurons as you choose parameters. This is what's well known, that you could actually capture a big variation in the qualitative dynamical type of spiking using a reduced order 2D equation, for example. But the epileptor is very, let's say, is tailored and cooked specifically to be efficient in capturing seizure events. And that's why you feel like, okay, why is this 0.002? Why is this 0.6? Why is this? So this is, it's very finely tuned and cooked to actually be efficient in capturing what was seen in the empirical data. So it's based on, so if what I mentioned is when the taxonomy of seizure types, they analyzed over 130 patients and over 1,000 seizure events, and they found the dominant type. And then they tried to cook up, what they did is that these equations were cooked up to capture that dominant type.
Yeah, but the calibering of these coefficients must be a huge task. I mean, this is not the caliber. I mean, to calibrate the coefficients like 0.002.
Physiologists probably have done such work, right?
I'm not sure. I mean, this is the epileptor was, I think, published maybe 7 years ago, but it seems like that. But then, you know, when modelers want to do phenomenological models, they take bold moves and for what it works. So this wasn't fit per se, wasn't optimized, let's say, to capture exactly. It was fit to give qualitatively good behavior, because we don't need much per se. We need something that's able to go into a fold bifurcation and out in a homoclinic bifurcation. And we needed to do it with the minimal type of algebra.
Yeah.
And so that's how this was designed. But if you'd like, I could, you know,
no, this is very interesting. I mean, you know, this is the first time seeing such model. I think, especially it touches, we had cases of epilepsy. You know, just to see it reduced to equations. This is very, very interesting. I think that's
but it is not. This is not explaining epilepsy. This is not the epilepsy. This is, for example, it's
an epileptor.
Yes, exactly. The idea is that this is, and I think that's the main statement here, is that if you identify the problem statement, what is this? So the VEP, the virtual epileptic patient, which is virtual brain built specifically with epileptor, was cooked for the specific purpose of helping in identifying the epileptogenic zone. So I would not take it, for example, and go test for the effect of drugs, epilepsy drugs.
Okay.
So because it was we have a very clear dynamical problem That is, we have a system that can be modeled as dynamical nodes on a network, and you have a very, let's say, clear dynamical event that's happening in the system. And from the physiology, we have a lot of already information about the mechanisms that are happening. And so the way it works is that, okay, can I use dynamical systems tools to cook up a very efficient way to actually create a mathematical object that can be fine-tuned and fit, personalized to patients, that can actually point me to where the seizure is starting.
Okay.
So that's why it's very, let's say, peculiar, but it's not in a way trying to explain the actual biophysical epilepsy process. So it's an engineering, let's say, solution that's based on dynamical systems in order to help surgeons identify the epileptogenic region.
Okay. Thank you. Thank you for your nice lecture.
Pleasure.
Well, any directions in also using some similar kind of modeling to for surgeries and other aspects of brain problems like in tumors and
Yes, basically it's happening. It's happening. This is so epilepsy, as I told you, this is already at the clinical trial stage, and but the framework is being developed to be much more generic, and you really need this interaction between the mathematician and the doctors to actually, because we know how to model stuff, but we need to know what's the feature that allows us to constrain and simplify the math.
Description of the.
Yes, so it's already happening. We're working on Alzheimer's. The models are being already developed. Parkinson's, we're putting in effect of dopaminergic pathways. There's work in stroke, multiple sclerosis, and so TVB has been already integrated also in something called eBrains, which is now been declared a permanent European infrastructure that's really also open source. So you could, you have data there, you could build your models, you could put in, for example, receptor maps, you could put in lesions, you could put, and then you could simulate and generate a synthetic idea. So yes, the framework is very, let's say, generic, and it's going in those directions for sure. It's not going to be, the epilepsy is sort of was the lower hanging fruit, because you see these bifurcations, and the system is going into a very, let's say, reduced complexity mode. As we said, it's increased synchronization, it's less, you see these big signatures, so you go into a very low dimensional dynamical regime per se. In the other pathologies, first of all, what is the actual mechanistic hypothesis that you will put in the system? For example, in Parkinson's, we put that in the model, you have these dopaminergic pathways that are damaged. We know that. We can put that in the model, but we don't have that clear dynamical feature, the seizure, for example, to try to use to constrain our model. So we're working on that. It's a more subtle notion. So each pathology needs to match, let's say, these two. But yes, it's happening. And if anyone is have any specific, let's say, disease or context in mind, of course, we would love to discuss.
So, quick question: would it be possible to, like in the case of Alzheimer, to start from EEG data and like, given the connectivity of the network, infer what the dynamics are, and like the interaction dynamics that you're assuming? So you just like discover all. So just having to discover what x0 is, you discover all the coefficients.
Yes, but we're not sure you would want to use the epileptor for Alzheimer, because if you're not going, if you don't see in your empirical data that you have this kind of seizure events, then why would you already put it in the in your hypothesis? But what we could do for Alzheimer's, for example, is we use more generic neural mass models, which is that there are some now which are you just have an equation for the mean firing rate, an equation for the mean membrane potential, by assuming that neurons can spike, you know, when pushed.
So I'm saying, what I'm trying to say is just assume a very generic
yes,
function of like
exactly.
polynomials and other functions, and then there are algorithms that sort of pick which terms are relevant and which aren't. So they sort of
Yes, but the
specify
problem is, do you think that these algorithms are still computationally feasible when you're dealing with such a high dimensional space?
So sometimes they are, because they're just linear regression algorithms.
Then you could, yes, the problem with that is going to be you could, yes, you could.
Yeah.
If you have the computational powers, of course, so this is actually what we're doing, except instead of starting with a blind polynomial expression, we start with a neural mass model where we know that if we cut off one region, we know what it's supposed to be doing.
I see, yeah.
It's supposed to be, you know, either a firing or so on. So we don't have to go in blind, because we know.
No, definitely better to constrain the model, yes.
But it's actually a polynomial at the end of the day. So this mean field model that I'm telling you is simply a quadratic failure, except that from dynamical systems, we know what what some of the coefficients need to be in order to already be in the dynamical regime that makes sense for a population of neurons. So so we start with that. We use personalized connectivity. We use personalized measurements, EEG, MEG, and whatever. We put in a mechanistic hypothesis about the pathology. For example, in Alzheimer's, we look at PET scans of certain proteins, the tau, the amyloid beta. We use that as a mask. And then we say, if this is what we know about the physiology and the pathology, fit those parameters for this patient so that I can have an avatar, which I can use as an in silico experimentation platform. This is sort of the general approach that's being done. Yeah.
I see. Yeah, makes sense. Nice.
Well, I think if we have no more questions, we can end our meeting at this level. Thank you, Hiba, very much. It was pleasant to see all this work, and also in the discussion at the end. Thank you, everyone, for those who raised questions. And we hope also to have more discussions during the upcoming workshop in October on different issues raised from the medical community to the mathematical community. Thank you again.
Thank you all. Pleasure.
Thank you.
Bye-bye.
Thank you. Bye.